Come on in. Well, thank you very much. I'm happy to be here. And my apologies for not being able to make it the last time I was invited to speak, but I was caught in a terrible traffic jam, as we've already kind of talked about a bit. But anyways, I would like to talk to you a little bit about children's brains and how they grow well, and how they sometimes do not grow well. But first of all, let me just perhaps say a couple words about myself and my background, so you know my strengths and weaknesses. And my background is really in clinical neuropsychology. So I'm a clinical psychologist who works with patients who have brain injury, and I teach graduate students how to assess individuals who might have a brain injury. And the sort of research I do partly overlaps with my topic tonight, but not 100%. Most of what I do these days comes from a new center we've established at UNM. Last year we got an $11.5 million grant to establish a new research center for brain recovery and repair. So this new center at UNM on the med school campus is designed to try and figure out some new treatments for people who have brain injury. So anyways, I think that's a nice addition to New Mexico and to UNM. But tonight we're going to talk about kids and kids' brains. And let me get used to this stuff. So why should anybody care about this stuff? Why should we care about neuroscience and kids' brains and how they develop and what might go wrong? Well, perhaps this is a question I can only speak for myself on, and I hope you folks are interested. But for me, it really hits on a lot of things that get me going as a professional, that get me excited and get me interested in my work. If you think about brain function and all it confers and its meaning in our lives, being able to provide people and ensure that our society encourages brain development is one of the best ways as people that I think we can help others achieve a high quality of life and help others achieve their individual potential by helping them grow their brains as best we can and have all the skills that they could possibly have. That's a way to fulfill potential, and that's meaningful for us all as individuals. I think there's a social justice angle here too. One of the biggest resources we have that we never talk about are all those resources that let our brains grow well. From healthcare to food to how we treat each other, and these resources that allow our brains to grow well are not equally distributed through society. And we need to pay attention to that. I also think, well, relatedly, that our intellectual resources, our capabilities as individual human beings, this is our greatest resource. It's not that we have oil in the ground. It's not that we have beautiful skies and great weather. We've got a bunch of people with great potential, and maximizing that potential is the way to maximize, one of the ways to maximize the benefits and level of our society and culture. Plus, I think it's exciting and fun and challenging. There's so much going on these days with neuroimaging that allows us to see what's really going on with brains, with genetics that allows us to try and understand the particular influences that lead brains to grow one way or another, that this field is evolving really quickly. And that's what makes it fun and exciting for people in the business. So what I'd like to try to do today, more generally, is first of all encourage you to ask questions. I don't have to go through this list of topics at all. Any questions you've got, I hope you feel free to answer, or feel free to ask. I'll try and answer at any time. So what I'd like to do briefly are these four major topics. I'd like to provide a brief review of brain function. Some of you might know much of this stuff, but I found when I try and speak to mixed audiences that some people know a lot and some people know none, and I'm better off sometimes bringing everybody up to speed with some basic reminders about how brains work. Then I'd like to focus more specifically on how brains change over early development. A little bit on prenatal care, but mostly on what's going on in younger childhood, okay? And you'll see I have some particular reasons for that. A word or two about adolescence, but ask questions if you like. So I'm going to talk about how the brain changes over childhood and what that means and what it confers for us in terms of improved brain performance. I'd also like to talk about the causes of variation in brain function. We all know this, you're teachers, and I am too. And we all know that there's variation in brain function amongst our students and amongst people in this audience, amongst people everywhere. Where does that come from? How do we understand this variation amongst us? And I'll be focusing on the variation that is problematic. Now, of course, we all vary, and some of us have wonderful gifts, but the problems that we have are the ones that we want to focus on socially. So we'll be focusing on some causes of brain abnormality in children. We'll focus on these sorts of variables. Neglect, abuse, poverty, and prematurity. There are many others, and I'm happy to talk about them too if you have questions. And I'd also like to talk about some treatments. This is perhaps the most exciting set of developments in my field in the past decade or so, that we have now developed scientifically proven treatments that help children in terms of brain function. Now, there are limits, okay? And it's not for everything and all of that, and there are lots of caveats. But the development of new ways to facilitate brain development in kids is really something that I find rather exciting. So anyways, that's the general goal. And if I'm going on too long on something you're not caring about, just wave me and tell me to go faster. And if you want me to slow down, tell me that, and please ask questions. So, in terms of a little bit of background on brain science, let's go through gross anatomy, cells in the brain, the idea of neural networks, and metabolic resources. So, let's take a dive in. Let's look under the hood and see what the brain looks like. So, the scalp has been opened here, as you can see, and underneath it is one of the meninges, the dura mater, this tough fibrous sort of thing. It's kind of like the inside of a football helmet that keeps your head from striking the plastic on the football helmet. You got that webbing inside. It's kind of like that. Then below that layer, oops, I don't know what I just did there. I pressed the down button. What does that do? Okay, well, it's back. This is another picture there. That's the brain with the entire skull removed, showing you the outer layer, the meninges, and beneath that, I'll get these buttons straight before I'm done here. As you pull off the dura mater, you see the surface of the brain here covered with an arachnoid layer, another layer of meninges, and beneath that arachnoid layer, another view, lies the brain itself. So here's the cerebral cortex, all you can see right here, full of gray matter, as they call it. And at the back of the brain is the so-called little brain, cerebellum, important for motor coordination and fluency. I would like to emphasize that this whole darn thing here is one organ. It's one thing, okay? It's like a liver that kind of sticks out here and sticks out there, but it's not like it's 12 different things, even though sometimes we talk about it like it's 12 different things, okay? So here are those 12 different things. We can kind of divide that cerebral cortex, that gray matter, up into all these different regions that do somewhat different things. The important thing to remember, though, is all these regions work together. Now, one of the things that I will be emphasizing as I talk further are the functions of this region, the frontal lobe, which tends to be more or less responsible for what we tend to call executive skills, and the parietal lobe right in here, which along with the frontal lobe, kind of the seat of intelligence, sort of, to a first degree, okay? So let me take you through a little bit more detail on what these different parts do. The temporal lobe is right in here, and this would be the front of the brain, and that's the back. Temporal lobe, hearing, emotional control, and complex visual systems. And I'm just going to go through this quick to give you an overview. The parietal lobe, which is, again, right up in there, which is important for integrating information from different sensory modalities, auditory and visual and tactile, for example. They get pulled together and compared there, so you can link them together. But the front part is involved in our sense of feeling or tactile perception, and a lot of spatial skills are there, located there too. Map reading and finding a route, things like that, okay? Moving along, here we have The frontal lobe, this 1/3 of the brain, it's huge. The posterior part of the frontal lobe, right in here, is responsible for movement and motor control. So the left side motor strip controls the right side of your body and so forth. Now, in front of that part, though, that is, in front of this posterior part of the frontal lobe, this part right up here is called prefrontal cortex, and we can subdivide that even further, and I'm not going to bother trying, but I would emphasize that these regions that we see in color here are especially important for what we'll talk about in just a moment as executive functions, a very important kind of cognitive skill. Now, underneath all of that gray matter, underneath all of that cortex, lies an incredibly vast array of connections, fibers that link different parts of the cortex, and this is the white matter of the brain. Think of it as connecting pathways linking parts of the gray matter. And that's just an old anatomy slide to give you the general idea, but here is one of the remarkable sorts of demonstrations of what we can do with neuroimaging these days. This is from a live human that this picture has been developed, showing you only the white matter, only the connections, the pathways that link different cortical regions. And I just think it's amazing. Look at it, kind of looks like, you know, somebody with blonde hair stuck the finger in a socket or something, right? And just all the hair just went like that. But it gives you an idea of the richness of all these connections, okay? As well as our tremendous capacity to visualize these now and try and figure out what might be wrong in someone with a disorder. Now, about those pathways, we can divide those pathways into many different types. And here are just 6 examples of particular pathways, white matter pathways in the brain, color-coded, and you can see where they are. So in this view right here, this is as if you were viewing my head from this angle, okay? Just take off my skull like that and look straight on. In this view in the middle here, it would be if you just like kind of cut my head like that and look straight down. In this view is kind of funny that it's straight back like that and then up. But in any case, you can see just for these 6 different pathways here, and there are many more, you can see how they spread throughout the brain. And I would emphasize too that people differ in these precise pathways. Some of us might have, just hypothetically, an especially large forceps major, which is important for communicating left and right hemispheres, and some of us might have an especially small superior longitudinal fasciculus. But these variations amongst us in how our white matter connects the processing centers of the brain, it has functional consequences. These differences amongst brains exert influences on what we can and can't do, our strengths and weaknesses. So that's your real quick view, gray matter, white matter, the centers that do the processing and the pathways that connect them. Now let's dive further down into the brain, though, at a different level of analysis, and think about the cells that actually comprise all that gray matter. Neurons are scattered throughout the gray matter, define the gray matter. That's basically all you got in gray matter, are cell bodies of neurons. And this is kind of what they look like. Okay, let me draw your attention to a few different things. So here's the cell body where the genes are and manufactures lots of stuff that gets sent around to other parts of the cell. And here is the axon, the long projecting pathway away from the nucleus of the cell. And here at the end are axon terminals. And right here at that axon terminal, for example, would be a connection to the next neuron in line. Okay? So we have surrounding the nucleus and the cell body here these dendrites. Other neurons would come in and communicate with them, would come down and form a synapse, which I'll define in just a moment, with these. And so you can link up one neuron to another, to another, to another, so that this cell gets input from every other neuron, hundreds and hundreds of them, that form synapses on these dendrites. And this one will connect with many, many other neurons from this one part and that part and so forth. So here is your basic computational unit in the brain, the neuron. A few things I'd like to mention about these cells. First of all, neurons in the cortex, in that gray matter, are basically all formed before birth. Technically, a few others come later. Don't worry about them, because they do next to nothing. So we are born with many more neurons than we need, many more than we'll ever have at any other point in our lives. The implication, obviously, is once you lose them, that's bad news, because they're not coming back. These dendrites here and these axon terminals here come and go. They're very plastic. They change. They change with environmental inputs. They change with stress. They change with age. They change with what you're trying to learn and what you're focusing on. And it's these changes in how dendrites connect to other neurons and so forth, changes in that underlie our learning ability. So one other thing, point number 3 here. The axon itself, this long projection here that goes from the cell body down there, is covered with something called myelin. It makes it work better. I'll tell you more about that in a second too. But myelination is progressive. This isn't something that's over and done, and occurs through early adulthood. And finally, I'd like to just say the brain development never ends. There is no end. You know, there's no maturity, there's no stage you reach and where that's that. The brain is responsive to all the things that are in your environment and all the things that you do, which changes throughout your life. So of course your brain changes too throughout your life. There is no static endpoint here. Well, we can think of a static endpoint, but that's not the one I mean. But anyways, brain development never ends. The brain's always changing, is always plastic, always adjusting to our circumstances, no matter how old we are. Now, when those dendrites and axon terminals connect, they do so at a synapse. So here we have, in a blown up example, this would be an axon terminal coming close to another dendrite right here, okay? And here's the synapse where so much goes on. This is what we really need to understand well to understand a great deal of learning ability and the changeability of the brain. Now, what happens in the synapse is that neurotransmitters, chemicals, are packaged in these little vesicles here, in those little cellular components. And when this axon becomes active, these vesicles move down towards the cell boundary and discharge their contents into the cleft, into the space, before running into this dendrite of another neuron. And basically, when these neurotransmitters affect this next neuron in line, they cause electrical changes that propagates a signal. Okay? This gets very complex, and I don't want to get into this in great detail now, but basically, these synapses come and go with maturation and learning and experience, and changes in synaptic function really underlie learning. That's the physical change in our brain that lets you know new stuff, is different patterns of connections between neurons due to differences in efficiency in synapses. In almost all drug effects you'll find for psychiatric disorders, mental health problems, etc., almost all drug effects and how they work are by manipulating this machinery right here, by altering how much neurotransmitter gets released, how quickly it gets degraded, the effect it has on the postsynaptic dendrite right here. This is where drug effects are to be found, and understanding the synapse is the key to understanding and developing new drugs. Now, with all that stuff on gray matter and white matter, I do want to emphasize for you that the real job of our brains, the real function, is done by neural systems or networks. That is, collections of brain gray matter regions linked by white matter regions. So any given function is accomplished by a set of interconnected regions or a network of gray matter and white matter. Just for example, here are 3 large-scale networks in the brain that have been described and really discovered only within the last 10 years. These are large-scale units scattered throughout the brain that perform particular jobs. So we have a large network that we some researchers term it a task-active network, and some researchers term it a central executive network. But this network becomes very active when we're solving most cognitive problems. If I give you a mental arithmetic problem to solve, this would become active. If I ask you to solve an analogy or something like that, this would become active. Ask you to remember a phone number, it becomes active. Another big network is called the default mode, which is, again, a large-scale network of many different regions scattered throughout most of the brain that seems to be especially active when we're doing nothing, when you're sitting and reflecting, thinking about your life, things like that. It's kind of interesting. Daydreaming, and so forth. Ruminating. And then there is this other network that is termed a salience network, or importance network, or what do I do now network, that is responsible for switching back and forth between the active task network when you need to solve a problem and the reflection mode when your default mode is more active. So when the flexible deployment of these two networks is determined by that one. Okay? So again, just a general picture on how brains are built and how they run, with a focus on networks being the real functional entities that do the job. Yes, sir?
How much uniformity is there in the brain in, say, in solving problems? In other words, if we take 10 people, how similar would their network use be?
If we assume that these are all people who develop normally and typically functioning adults, there's not a great deal of variation. Now, to the extent that you or I might have unusual experiences in our background, and let's say that we are both interested, or you're an artist and a visual artist, and I'm a musician or something. Now, with that extremes, you might see some variation in how people process these things. But in reality, there's not that much variation. In fact, in these networks, for example, can be demonstrated in prenatal humans. They can be demonstrated in other primates too. So, more basic stuff. So again, I want you to think about networks as being the organization unit that gets the thinking done, okay? And so think of a network as something like this, where you have a bunch of different brain centers, illustrated in blue here, okay? And then connections between them in green. And then we can kind of extract that network from the physical brain and think of it as a more abstract graph like that, in which we have what in the parlance of graph theory, these would be nodes or centers, and these connections or pathways. But what's nice, what I find interesting about this work is that we can now bring a lot of the mathematical language of graph theory and topology to bear on understanding how networks function. So graph features that are relevant, for example, understanding networks. You could think of this as power stations and transmission lines. And the formal properties of a power line transmission network system are the same as the brain. So anyways, I guess I've said enough about that. But think about the functional organization of the brain as network-driven. I'd also like to mention one last thing about basic brain function that I think is underappreciated in neurosciences these days, and that is the metabolic demands of the brain. The brain uses so much energy, it's amazing. And we need enough energy to run it. So what does the brain run on? More or less, the brain runs on two things: glucose and oxygen. And that's that. And since the brain manufactures neither of these, they got to be brought into the neurons from elsewhere to make things work. So if you don't have enough glucose, well, that leads to hypoglycemia, and you may have run into or heard of the problem the hypoglycemic individuals may have when their blood sugar gets really low. They get confused and kind of can act like they're not fully with it, as if they were intoxicated or something like that, solely due to the fact that they're not getting enough fuel for the brain. And of course, oxygen is necessary too. 4 or 5 minutes without oxygen, ooh, bad news. And some of the common causes of oxygen loss are carbon monoxide poisoning. We see way too much of that in New Mexico. I've seen a lot. People with really bad heaters that fill up their house with carbon monoxide at night. And what happens is the carbon monoxide binds to the hemoglobin molecules in the blood preferentially and binds stronger than oxygen does. So it effectively eliminates oxygen carrying capacity. Heart attacks can do that to you too, obviously. And some regions of the brain need a lot of resources, a lot of metabolic resources. In particular, hippocampus, which is very, very important for memory, and a set of structures called the basal ganglia, which are really important for initiating movement and initiating and starting cognitive activity. So when I see patients, for example, who've had some problems with glucose or oxygen, these are the brain regions I test first because I suspect they'll be most affected. These are the brain regions with the highest metabolic rate, so they get affected first by metabolic abnormalities. And finally, the brain uses an incredible proportion of our metabolic resources during childhood. I think this is an amazing statistic. 70 to 80% of a child's metabolic needs are for the brain. That's amazing, isn't it? Yes?
What age is it shift from 78?
I got a figure for you here. Obviously less than adulthood. But let's look at this figure here. So here is, I'm sorry this isn't a very pretty picture, but but anyways. So here we have on the x-axis just age of the kids, and on the y-axis is glucose utilization. Just think of the y-axis as metabolic resources. And it goes from 0% to 70% here, okay? So this is the proportion of all of our metabolic resources that go to the brain as a function of age during childhood. These are, the top line are boys, bottom line girls. That's interesting in itself. But look at age 5. Look at how much, in this study anyways, of our metabolic demands are for the brain. I mean, think of it. Look at a 5-year-old. How much of their body is brain? It's a pound or two, and yet it's using all that energy? Well, yes, that's true. So here's an interesting observation. Yes, sir?
So you differentiated boys and girls. Which one goes on top?
Boys. Greater demand in boys. As befits the greater vulnerability of boys to most neurodevelopmental issues.
Do they have a proposed reason why they think boys?
This is for the sex difference?
Yeah, why?
Yeah, it's a long answer, but the basic idea is that for lots of different variables, whether we're thinking brain function or other aspects of physiology, there is greater variation amongst males. There's greater variation in height. There's greater variation in weight, in brain size, in intelligence. Greater variation, not a mean difference, mind you, but in variation. And this stems, some researchers would argue, from the fundamental asymmetry between men and women from an evolutionary perspective, that women are choosy. And it makes sense for men to disperse in sort of phenotypic space so that many different varieties of guys are out there. So women have somebody to choose, have a range. I know, but that's a little bit far-fetched, but it's the only explanation I got. But anyway, I do think you need to turn to evolutionary issues for explanations like that. But here's, oops, where am I? Next slide. So now I've also, in addition to the regular figure here you kind of saw, which is the same figure for metabolic demand, although on a different scale, here's what happens to weight gain during the same period. Now, notice then that at this point, where the metabolic demand for the brain is the highest, there's very little weight gain going on. There's so much energy demand for the brain that the body can't grow. So I really think, and I'd like to emphasize, that I think this is a vulnerable period. If you're on the edge of your energy budget like that, you're at risk. So, quick view of brains from structure to function and metabolism. Okay, I'm going to now switch to talking about some things that happen in brain development, mostly in childhood, but I'll give you a larger picture earlier on too. Okay? So, if we think of what happens from conception on, these are the basic stages. First of all is neurogenesis, or cell formation. This happens quite early in fetal development. This is first trimester stuff. After the neurons are formed, they need to migrate and move to the correct location. If that doesn't happen, we have problems. These cells that are generated very early in fetal development will need to differentiate into a variety of types of neurons. Some in the cerebellum, and some here, and some there, and the different shapes and do different things, different varieties. Cells will grow and mature, and this is where you see explosion of dendrite growth and axon growth. Next, we see the development of synapses. And everything before this line here is prenatal. Okay? You also see a good deal of cell death prenatally. So many cells are generated, we don't need anywhere near that many. And the process of building a brain is more like sculpting than adding step by step by step. So you start with something, you generate something big, and then you sculpt away the parts you don't want, as opposed to you take a little thing, and then you put a little bigger thing on it, and a bigger thing, and build it up like that. So, and these things are mostly after birth. Let me give you a little bit more detail and some more pictures on that. So here we have a nice figure on how to build your brain, starting from conception on through birth, right here. And again, neurogenesis and neuronal migration occur very early, okay? And these are the stages this is all fetal, fetal development, age of gestation. It's right in here where these major processes are occurring of migration and neurogenesis. If there's a problem here this early in fetal development, it's going to have a major consequence, okay? Because this is sort of the foundational start of the brain. And so if that doesn't get going, you're going to have major difficulties, including, well, mental retardation or things like fetal alcohol syndrome have their effects by influencing fetal development at this particular point in development. And those effects can be quite severe, as you know. So that gives you a picture of what happens before birth. And I also wanted to show you some pictures of individual neurons and what they look like. This is from the prefrontal cortex, from a newborn on through 2 years. So in the newborns, you can see, well, here's one neuron, and there's another one, and another one. And you can see how sparse these things are, right? They're not bushy or anything like that. They don't have much of dendrites. They don't have much of axon terminals. But that stuff all grows and happens very fast. So by 2 years, you've got this rich network of neurons with very dense proliferation of dendrites and forming synapses with each other. So there's a good deal of a lot of that maturation happens quite early. Now here's a picture that gives you a sense of how brains develop in terms of overall features, like overall size. So at very early in the first month of development, the brain is simply a neural tube that looks like that. What I'd like to draw your attention to is the fact that at 9 months of age, it looks so much like an adult brain. And by age six, a kid's brain is 95% of adult size. But at age 6, I don't know about your kids, but at age 6, my kids weren't doing too much. You know, they're smart kids, but it's not like talking to my wife, you know? And so what I'd like to emphasize now is that so much cognitive development takes place after age 6, right? I mean, we all know this, but it's not because your brains are getting bigger. That's not why that cognitive development occurs. It's not just getting bigger brains. We got to deal with the quality of the brains to increase our skills from age 6 on. And how do we do that? 2 major processes sculpt and organize children's brains as they go through birth through 5, 7 years old, things like that. And let me emphasize that our genetic plan for development is only kind of a rough sketch. We have so many connections between our neurons and so many neurons, we cannot have genes code for individual connections, or this one goes here and goes there and so forth. It's the plan that's loaded in the genome with particular algorithms that allow sculpting. So what we do first is generate a gazillion synapses. Synapses everywhere. Lots and lots and lots of connections between lots and lots of neurons. But only those neurons that fire together can survive. If this one fires and is active and ignites or excites this next one in line, these two will both live and you know, persist through adulthood. But if this one's active and it doesn't get this one active, this one's going to go away. So we end up doing what this is also called activity-dependent pruning, so that the parts that are working, that are forming networks, that's what we keep. The other stuff, don't need. Let's get rid of it. And it's that process then of removal of the waste that makes brains efficient and gives them, in part, their better cognitive abilities as we age. As we'll see too, different brain regions develop on different schedules. And this isn't happenstance. This is an adaptive design of our species. Here, for example, we can look at how the visual system develops on one time scale, and prefrontal regions, important for executive skills, planning, organization, etc., develop at a different pace. So here we have on the y-axis, the density of synapses, and on the x-axis, time. And you can see that for the visual cortex here, it peaks earlier and then declines earlier. And the curve for the prefrontal cortex, while it is generally matches the other curve for the visual world quite well, it's kind of shifted to later in development. Well, that makes kind of sense, doesn't it? I think so, anyways, that the visual cortex is kind of an invariant feature of human lives and human existence over time, over eons. Our social worlds change all the time, right? And your social world is different than mine, perhaps, but our visual worlds are pretty much the same. So it makes sense to have some systems that we want fine-tuned to our environment, our individual environments, develop later, so they could be shaped by those subsequent environments. This slide just kind of shows you some of the changes you see over age, and that the loss of synapses and dendrites and this pruning process makes the cortex thinner as you get older. You might think it the opposite, that as we learn and get more capable and grow and study stuff, our brains get bigger and bigger like a muscle does. No, that is not true. The cerebral cortex, in fact, shrinks and becomes thinner as we age, and this figure shows you that. So the blue areas are getting thinner and red are getting thicker. And you can see by the time you go from 5 to 20, you got a lot of blue. It's thinner down here in general than it was back there. So this is not a case of bigger is better. This is more efficient is better, okay? So pruning of synapses and dendrites, that's one big sculpting process. Here's the other process in childhood that's critically important to make those brains work better, okay? To allow them to become more functional after age 6 without getting bigger. Myelination, and this is the process I mentioned earlier, that glial cells, or not a neuron, but another type of cell in the brain, glial cells, wrap around the axon and make it work better, okay? It's kind of like, this is only an analogy, but it's kind of like an insulation on a wire. Insulated wires conduct better. Myelinated axons are more efficient and work better. But it's not exactly like that. So anyways, this myelination process, which makes your white matter more efficient, goes on for a long time. This is not something that is done when you're 6, 7, 8, etc. Definitely, white matter changes and myelination increase through the 20s and maybe the 30s. So this is, again, the point that the brain is not static. It's always either something's growing or you're losing it. It's one or the other. Nothing stands still. But anyways, better myelination leads to faster processing, faster signal processing, faster ability for one neuron to excite another, and it also allows more synchronized activity in its region. The rates differ across regions. But anyway, so here's a picture again of an axon, and this is the process. It's all this green stuff here is the myelination. And if you look at it down here, you can see it's kind of, the cell wraps around and around and around and around. You know, you just think of this as a sheet wrapped around the neuron a few times. That's the myelin, okay? And the quality of that myelin in part determines the functionality of that axon. This just shows you, as a function of age, increases in white matter for men and women. Slight sex difference there, no big deal. And this figure just kind of highlights some of the changes we've already talked about, but I would like to just draw your attention to neurogenesis, cell proliferation, the myelination, and these curves all represent the experience-dependent synapse formation. So sensory motor stuff comes early. That's the physical world we all live in and share. Parietal and temporal association cortex might be more important for language development, and prefrontal cortex, again, for executive functions and kind of social connectedness. The areas in blue are those that myelinate early, and those are kind of sensory and motor regions, because again, our sensory motor worlds don't much differ. And the things that myelinate late are those regions that are more important for adapting to our current environment that could differ amongst us. Here is the, I'm sorry, looking out the audience, this is bad news for lots of us, I'm sorry, that we are past this stage, this point right here, and we're into the degeneration stage of our myelin as opposed to increasing. But again, nothing static, right? Yeah, but I'd rather be on that side of the curve, I'll admit it, yeah.
I mean, these curves, how are they related to nutrition's other status? Because is it something that the shift can be moved further to the right?
I doubt it. I do not know. I mean, there's a difference between, well, absent deprivation, absent starvation, and deficiencies in vitamins and things like that. I don't know of much that can vary, that can affect the retention of myelin or not. Age is just the big issue, yeah.
Does myelination happen on its own in the brain, or are there things in our environment that encourage more myelination?
That's a good question. In general, a rich, stimulating environment encourages myelination, okay? Beyond that, I don't know what to say, though, beyond general good health and good genes and stuff. But there is definitely, you can see this from animal studies, more clearly, as you can imagine, than human studies, but placing in an enriched environment is one of the things that boosts it. But you might know, too, that many disorders, more in adults, can affect myelin, like multiple sclerosis. This is the fundamental problem with multiple sclerosis, is your loss of myelin.
Yeah, that's one more.
Sure.
It's real interesting to look at. There's so many different factors that contribute to normal or healthy brain development, and that strengthen the connections between the parts of the brain and the different networks. And it seems that you'd be able to derive some sort of, pedagogical and/or environmental stimulus that would facilitate development of myelination or increase in synaptic plasticity or whatnot to, encourage more complexity in the brain that maybe you could, put it into schools, for example, or suggestions you might make to parents on how they raise their kids. I'm just wondering, is is the thinking around cognitive development relatively recent in terms of making these kind of suggestions, or have people been working on that?
Well, you're absolutely right in where you're going. That in the thought that, but the, kind of what I've tried to lead here to, the fundamentals of how this is done does lead to some optimism about things that one could do to intervene. And you know, we can jump to that sooner or later, but I completely agree with that. But you know, it's a parametric question, sort of how much stimulation leads to how much improvement in white matter, leads to how much behavioral improvement. And while things can go in that direction, how far do they go? And the answer is not enough to cure a lot of the problems we'd like, you know? But absolutely we can affect brain development with interventions, behavioral interventions now. And I'll get to some of those, because I think that's perhaps the single most important thing we could talk about. All right, away from this bad news. So I wanted to, one last slide here on this stuff. So as pruning and myelination take place, these networks get stronger. And I guess I will stop right there, but we can empirically demonstrate that the connections between different regions, outlined here for example, become stronger in adults than in kids in each of these regions. And it's these changes in the efficiency of the networks, again, that confer the behavioral advantages of older childhood and adulthood. Now here's my one slide on adolescence. I wanted to focus on younger issues, but there's an awful lot of interest in adolescence these days because of the association of adolescence with risk-taking of various types, especially substance abuse. So here is the challenge of adolescence, at least the conventional explanation of it, that parts of our brain that are especially sensitive to reward in the parts of the brain known as the limbic region, that is sensitive to rewards like food, sex, money, drugs, these areas mature more quickly than do the areas that control them. So we have very sensitive reward areas, exquisitely sensitive to social rewards like status, which of course we know adolescents, you know, pretend to those things. And that the prefrontal cortex here matures more slowly than these limbic regions, and the prefrontal cortex really is important for inhibitory control, to be able to delay gratification with respect to reward. So it's this gap here between our demands for rewards and our capacity to inhibit in socially appropriate ways deal with these things. It's this discrepancy that yields the risk of adolescence. And this is thought to be one of the major sorts of reasons why there's an increase in the incidence of substance abuse, risky sexual behaviors, and things like that during adolescence, is the differential growth rate of these two critical brain regions. But I want to add that this is not bad. This is not like a problem in the sense of brain design. This was how people were built. We were designed this way on purpose because this is what in ancestral environments sent adolescents out on their own way. That was necessary, obviously, from an evolutionary perspective. So I'm going to move on and talk now a little bit about a couple particular cognitive skills that I think are really important, that are very different, though, in some ways. We all know what intelligence is. Here's the conventional definition by the guy who invented the test, David Wechsler. Intelligence is the global capacity of a person to act purposefully, to think rationally, and to deal effectively with their environment. Okay? So that's one skill I want to talk about. Here's the other skill I've mentioned several times already, executive function. These are positively correlated, but independent, different growth curves, and executive function is more malleable, but I'll get to that too. So what is intelligence related to? Oh, geez, here's a partial list, most everything. Individual variation in intelligence is associated with such things as personality, brain morphology, social outcomes in terms of mating, education, occupation, socioeconomic status. Intelligence is linked to risk for different forms of psychopathology, schizophrenia, depression, dementia, substance abuse I didn't have up there. Intelligence is linked to health in terms of morbidity or illnesses and in terms of mortality. Yes, believe it or not, smarter people die older. They live longer and have fewer health problems. And interestingly, it's not just because they eat more salads and stuff like that and know more about nutrition. That does not appear to be the case. Does not appear to be the underlying reason here. It's probably more a fundamental issue of how the brain and body was built at the outset. But here's one thing I want you to know about intelligence, and which is different than executive function. It's remarkably stable. Here are in the scatter plot here, performance on a good but kind of unusual intelligence test at age 11, and then the same people, believe it or not, were tested at age 90. That's a rather remarkable data set. But you can see that it's relatively stable. People don't change too much, though they can. I don't want to say that it is fixed, but it's kind of fixed. It's not that malleable, okay? Let's talk about executive function a little bit more, because you probably have heard less about that than intelligence. Executive function, which is sort of the province of the prefrontal regions I mentioned earlier, broadly thought of as planning, organizational skills, how well we respond to unstructured environments, how well we can control and regulate our emotional responses, stuff like that. But here are the three big components, according to most kind of definitions. Inhibitory control, working memory, cognitive flexibility. Let me give you some more details on those. So here's some of the inhibitory control aspects of the frontal regions, executive function. So what I want you to do is take this test. Your job, you kind of have to do this. I would ask you to do this out loud, but it would be, the cacophony would be disrupting. But anyways, I want you to go through this here and not read the word. Do not, to yourself, say blue. You should say green, purple, yellow, red, etc. So I want you to start here and to yourself, just to yourself, go through and to yourself say the color of the word, not what's written. So start here and go on through to here. All right, enough. Now, okay, now I want you to start here and just read them and go through to there. Just read them, words to yourself. Blue, red, etc., as fast as you can. There's a difference, right? I mean, you got to be able to see that difference. And we have to learn this inhibitory control, okay? That's an example of an executive kind of skill. And learning this inhibitory control is the same thing as learning to inhibit an emotional response when you annoy me, but this is not the time for me to have an argument with you, you know? That kind of thing. So I'm gonna wait, and when this is over, we'll talk. You know, that kind of control is similar, yeah.
Is there a relationship between this and something like implicit bias?
There is, but only in terms of the methodology used to assess them. I don't think implicit bias is something I really want to throw into executive function, but this sort of slowed responses you showed when you tried to name the color, yeah. That's the method that detects the implicit bias type stuff. Here's another aspect of frontal lobe function. So you see that arrangement of discs here on the pegs up top? That's your goal. You're given this. Now, what I want you to do is move one disc at a time, and you can never put a bigger one on a smaller one, and go from this setup to that in the minimum number of moves. I know I gave you a day you could do this, but I want the minimum number of moves as quick as you can. Well, there's some planning there. You see what I mean? I mean, you move this there and you move that there. So I'm trying to give you examples of executive function. This is what we're talking about here. Executive function, like intelligence, is linked and associated with lots of stuff. It's linked with vulnerability to schizophrenia. Low executive function is a risk factor for substance abuse. High executive function is what's often thought of as resilience. We all know that some children placed in horrible environments end up okay, right? They don't all end up bad, you know? Why do some kids survive? Well, good executive function skills are one positive reason for some kids having greater resilience than others. Lots of research shows better executive skills leads to school success. That doesn't surprise anybody in this room. Executive functions have also been linked with the current hot topic, grit. You know, same idea. I like executive functions a lot better. I'm going to skip this. Now, I'm going to skip that too. So what I'd like to do now, I guess, is summarize a couple points we've made and then move on to some different matters. But there's little change in brain volume after 6. So our cognitive development through childhood and adulthood is dependent upon these processes of pruning and myelination that shape networks and increase their efficiency. Okay? Now, what I'd like to do is turn to a different question here. Where do differences amongst us come from, and what can we do to help? But before I do that, I'm going to pause because I don't have a watch or anything. I got no idea what time it is and how we're doing here.
7:07. 56 minutes.
Okay, well, somebody just wave and stop me when you want me to stop or anything. Okay, let's think of first, most broadly, what are the different sorts of causes of brain abnormality we see in kids? And here's a kind of nice list. Of course, genetic abnormalities will lead to different kind of problems. Problems in early embryonic development, like exposure to alcohol, prenatal disease, infections occur prenatally too. And all of these things would be, well, these things would be worse the earlier they are in terms of the consequences. The ones I'll focus on in red here, birth trauma, an oxygen at birth, or really premature birth, which can lead to such things as cerebral palsy. Fortunately, malnutrition is not that common, and our brains are relatively resistant to most nutritional changes in childhood. Good design feature, but severe malnutrition has undoubtedly effects on brain development. And of course, we can have environmental abnormalities, that you can be raised by some people that are pretty abusive. That happens, unfortunately. One could be raised in a harsh and unpredictable environment, and one could be raised in a wonderfully supportive, enriched environment. And those things make differences too. Here's one point I'd like to make, though. As we think about, let's think about this notion of deprivation, and that some kids grow up in circumstances that are less than others. We know that exists. So think about it as a continuum here of environmental quality. With, on one end, gross neglect and abuse. Just, that's self-explanatory. In the middle, mere poverty. Poverty does not have to be associated with either neglect or abuse, but it is nonetheless a factor in environmental quality related to such things as healthcare, for example. And finally, more just simple normal variation. What I'd like to emphasize for you is that the function relating environmental quality to either brain status or ability, any kind of ability, is probably not linear. It's probably something like this. It's probably curvilinear, where most of the effects of environment are limited to a part of the distribution. Here's another way to look at that. On the y-axis, IQ. On the x-axis, an overall measurement of environmental quality from a very good researcher. So this is kind of like socioeconomic status plus healthcare, if you will, okay? And what Turkheimer showed in this data was this shaped function, which is really kind of interesting. It suggests that all the business is down below that point, right? Here's another simpler way to look at this. There may be a threshold, an environmental threshold, and above this threshold, increases in environmental quality don't much matter. And it's below that threshold that we see all the adverse effects. Yes?
Hold a second, so change tapes. Thanks.
Okay. So again, I was suggesting that the quality of the environment is indeed important for the development of children's brains, but it is probably not a linear function. That for IQ, which is represented in the solid line here, it's probably the case that once you get above a threshold, it doesn't much matter. And for those of you who have background in evolutionary psychology, this makes a good deal of sense. It has also led to the idea that, kind of good enough parenting or environment and so forth. So there's no need to buy your kids Einstein toys. They don't work, There's not much you can do. Once you get above the threshold, you're not going to do much to affect IQ. Ain't going to happen. But if children are growing up below that threshold, there's a hell of a lot we can do, and that's where resources should be placed, I would argue. Oh, one last thing I wanted to mention on this slide too is that the shape of the function for executive skills is probably different. The threshold is probably further up, and I'm not sure it asymptotes quite as much. I'm guessing here a little bit, but that's what I think, okay? So let me now, with that general background, let's look at some horrible environments. What are the effects on people? Here is about the worst sort of environment I can think of. The worst example that I've seen in the scientific literature, what happens to kids that were raised, my example is going to be in Romanian orphanages during the 80s, which was just god-awful, horrible environment, okay? Here's just a picture of where the babies were kept, and I don't need to regale you with the horror stories, I hope, from maltreatment to kids being left alone in unchanged diapers for months, and nobody picks them up, and just use your imagination. It is that bad, okay? Well, let's look at a study of these kids, okay? And we'll have 3 groups of kids we're going to compare now in terms of, first of all, intelligence. Kids that were moved from these institutions to foster care, okay, good foster care, those that were in the same old care as usual bad institution, and kids that were never institutionalized, okay? 3 groups. Here's what the data look like, and let's look at full-scale IQ over here. Now, 100 is average, okay? None of these groups got to 100. But the group that was the best care here, the kids that were moved out of the orphanage into this group, had a slightly higher IQ than those kids that were in either the original institution or another somewhat less bad institution. So the point is, I think the take-home message is mostly that, well, these kids did suffer an IQ decrement from that horrible environment, but being placed later in a nice adopting home can slightly ameliorate that effect. It doesn't normalize anything, not even close, okay? Here is where the effect is most profound, though, these differences in early environment is in terms of verbal abilities, rather than here is the column that's perceptual reasoning or kind of spatial skills. So again, the point is just that, yeah, there is an effect of these orphanages, okay? That level of neglect, that's on that left side of that curve, below the threshold, and by God, there's an effect. And intervention is only, even if it's a pretty damn good intervention, like putting these kids in really nice families, only had that much effect. An effect, but not a big effect. Now, what we don't know is how well those gains will persist as these kids get older. The gain to be had from being placed in a high quality environment. They may backslide, we don't know. Now here's a subsample of that same Romanian orphanage study, okay? A smaller group of them. These kids were given neuroimaging to look at their brain volumes and stuff, okay? So a smaller group. Same general idea. They were 10 years old. And here's in terms of gray matter. Care as usual in the bad foster care, good foster care, never institutionalized. Never institutionalized. That's good for you, obviously. But these two didn't differ. Okay? These early influences are generally kind of lasting. And here's on white matter, which shows a somewhat different picture, that here, for white matter development, that foster care, the good foster care, did have a benefit. Okay? So we're seeing some benefits from foster care, but they're not much, and they certainly don't ameliorate the whopping deficits that these kids have. I'm going to skip some stuff here. Well, those orphanages are about as bad as it can get. How about just families around, like in the United States, that are abusing their kids, either physically or emotionally and so forth? What does that do? Well, it mostly affects the brain's stress systems, the hippocampus. This hormone has little effect. This abusive family environment has little effect on intelligence, but it does have a modest effect on language and working memory, and major on the hippocampus and also frontal lobe sorts of stuff. Now, here's one large study I want to mention on the effects of poverty on brain development that is important for one major reason. I'm going to skip some slides because I know I'm kind of going slow here. I guess I'll go back to this one. So here's the model this study tried to test, a statistical model of relationships. They thought that poverty affects brain volumes, so poverty would lead to reduced brain volumes by virtue of mediating effects on parent education, supporting parenting, and child stressful life events. So in other words, these are the specific aspects of poverty that were thought to mediate the relationship between poverty and brain variables, okay? The way it worked out in their empirical results, we can just perhaps concentrate, well, look at either the top or the bottom one, but both of these are looking at how poverty or an income to needs ratio affects a brain structure. This is the hippocampus there, the big version here. But I highlight this box here associated with parenting because what this means is that it was the parenting, not parent education, and not childhood stressful life events, but how supportive and or hostile the parents were. That's what was important in how poverty affects kids' brains. It's not poverty per se. I mean, a kid 2 years old doesn't have any idea how much money their parents have, right? What they know is how they're treated by their parents. And thus, by knowing then that poverty exerts its influence largely through parental behavior, that's what we should intervene with. That's where our window is. And that's what we should do. So let me tell you about a couple studies that have been done to try and boost kids' brains and boost either one of these two skills I've been talking about, intelligence or executive function, okay? Here is the biggest project that tried to boost IQ, the so-called Carolina Abdisonarian Project. And this was a monumental effort to give kids a super head start type thing, and every kind of intervention you could imagine. They were working with the parents, going into the homes, nutrition, and parental communication style, and homework, and taxi rides to school, and everything you can think of. And that intervention did boost IQ for a few years. By the time the kids were in high school, it was gone. Okay? But interestingly, just came out in Science a little while ago, that those kids, although this treatment, this intervention did not boost their IQ, there was a much lower incidence of metabolic disorder in that group. That is, diabetes, high blood pressure, and so forth. That's interesting. But again, it goes to the point that it's really hard to manipulate IQ, intelligence. So I kind of think of intelligence is kind of like height, that it's very influenced by genetic variation, follows a growth trajectory set early in life. You know, most of people who are really tall were also tall as kids. And very generally reflects childhood health and prenatal environment, just like height. And interestingly, like height, it is also related to late life vulnerabilities. So the incidence of Alzheimer's disease is greater in those of lower intelligence, for example. It's not very easily improved. Let me tell you about this study, because I think I'll stop on this one too, because I just want to emphasize for you that there are things we can do. This is a study that's a little bit different. It's not a psychosocial intervention, but for very preterm birth, which is becoming more and more of a problem because more and more kids now survive being born at 26 weeks or 28 weeks or something like that. And if you're born really early like that, some of them, rarely though, fortunately, have cerebral palsy, mental retardation, very severe conditions like that. But there's growing awareness that the majority of children born this early have more subtle problems with academic functioning, intelligence, executive functioning, etc., that persist throughout our lives. Well, anyways, in this study run by Robin Olds from UNM, a valued colleague, we treated very preterm infants with either of these two drugs, which is basically, think of it as erythropoietin, which is blood doping. This is the same thing that marathoners got in trouble for a few years ago for blood doping, but this is what it is. So we gave EPO, abbreviation, at birth to children who were born very early or very small, and we're still following them and have been, and now I'll tell you about what's happened at age 3.5 with these kids. So here we've been able to show one of the very, very few interventions that increase IQ. Okay? So here we have kids that got the placebo. These are the ones that got the drug. You can see the mean there is better than that mean. That's significant. But here are typically developing children, TC, typical children. And we can't get this treated group up here. They don't get that good, but significantly better than the placebo group. That's for full-scale IQ. No effect on language, 0. Good effect, big effect on executive function. So these kids that were treated with this drug, come age 3.5, have better executive skills than those who got the placebo, and they were almost as good as healthy controls. And that's a clinically significant effect that would, if you can improve people's IQ by a few points, you increase the chance of going from a high school graduate to a college graduate, you know? Decrease the chance of mental illness. Further, we've been able to show that in terms of behavioral problems, like adaptive skills, acting out, other just behavioral problems in general, depressive type symptoms, that the kids given the drug had fewer behavior problems. That's best exemplified in this panel here, but there's an interesting effect. So here's the placebo kids, okay, from low and high SES backgrounds. And here's the kids that got the drug from low and high. If you're from a good environment, the drug didn't boost you. But if you are from a poor environment, those that got the drug have fewer behavior problems. This group has fewer behavior problems than that one. So technically, this is an interaction of SES with drug administration. So for IQ, everybody benefits. For behavioral problems, it's only those from lower SES group that's benefited. But that's still a big effect, and I'm happy to be able to report this, and I hope that it becomes used internationally. Let me just give you one other study. I know I'm going long here, but here is a study that is, I think, a model for interventions. This was reported in Proceedings of the National Academy of Science down at the University of Oregon, and they took kids 3 to 5 years old that were low SES kids in a Head Start program, and then added stuff into that beyond the Head Start, okay? So they got 8 weeks of training, and their parents got 8 weeks of training too. And the parents were trained in stress regulation, being responsive to their kids, providing them with information and resources. And the kids were trained on particular kind of tasks that encouraged vigilance and goal setting and monitoring your own performance and executive skills in general. And here are the parents benefited Okay? The parents who got the treatment showed more turn taking, less stress. The kids benefited, fewer behavior problems here than in these other groups, more social skills, changes in kids' cognition, language skills developed, nonverbal IQ developed, not verbal. Verbal is tough. Remember from the orphanages? We couldn't help their verbal skills there either. So anyways, this is just a behavioral intervention. This is taking parents and working with them, taking kids and working with them, and improving both the parents and the kids, and improving executive function, which again is related to school success, vulnerability to behavior problems and disorders, drug abuse. Okay? These are things we can do. So I'd just like to close there and emphasize a couple points. One is, yes, interventions will work. They won't be cheap, and they should be administered, though, to those who need them most, which are those at the lower end of that figure, the tail end I showed you, below the threshold. So we need to really target the right population and use our resources in that way. So anyways, there we are. That's all I got for you.